Christian Chojnacki. I'm a student at Interlake High School. My project is under lab research. Title is Investigating Gallium's Effects on E. coli and Bacillus cereus. So, my project was about seeing how does gallium actually affect different bacterias or bacteriums when placed in their environments. My original reasoning for this was to see if gallium was in fact, or if not, harmful in any way towards bacteria, and if so, then if it would be harmful for us, and if in commercial use it should not be placed. Additionally, if it proves to be antimicrobial, then it could be used in something similar to gallium copper pipelines, in disinfectants, coupled with perhaps even antibiotics to help eliminate the threat of bacteria in our society. So, my reasoning for my hypothesis was gallium is very similar to iron in atomic structure and very similar in how it reacts. However, the key difference between iron and gallium is that iron is redox active, which allows oxygen and enzymatic reactions to occur. Oxygen is allowed to be transferred in molecular structures like as hemoglobin, which is widely used in eukaryotes. However, gallium is redox inactive, which would inhibit oxygen transportation and or the enzymatic reactions from occurring, which would make gallium an inhibitor. Additionally, the protein ferritin that stores iron into a non-toxic form, which allows regulation for use in the cell, would be unable to store gallium, which then gallium would accumulate up to toxic levels and thus ending in the death of the cell. And it would also inhibit enzymatic reactions that iron would initially be a cofactor for, and then the cell would be unable to complete those biological processes created by iron or used in iron that would ultimately lead to the death of the cell. So, for my research, I decided on two separate experiments to try. One was to see if gallium was in fact absorbed into bacteria, and another was to see how does this gallium react when placed in the environment towards other cells, bacteria cells. What I found with my setup of the LB broth experiment, where gallium was placed in liquid state at the bottom of Falconer flasks or test tubes. LB broth was placed in afterwards. The bacteria were incubated in these tubes, and then it was allowed for light shaking and proper temperature for incubation. What I noticed was that with the corresponding levels of gallium, I have a graph here to portray that gallium was in fact absorbed by, that the wavelength absorbance increased with the increased amount of gallium in the tubes. And additionally, under microscope examination of the bacteria cultures, in both cultures, the ones where gallium was present, it was obvious to see that gallium was mixed in with the bacterial cultures, even though the gallium was not placed directly in the culture, creating a much more visible bacterial culture than normally seen in the controls. This proved that gallium was being absorbed. What effect gallium had on the bacteria remained to be seen. However, by seeing the bacteria streaked with gallium, I could further hypothesize that the gallium that was absorbed may have killed the cells, because those cells that were streaked with gallium were found near the bottom of the tubule, which may indicate that the cell, when it took it up, died and was sunk to the bottom of the tubule. My further experiment was incubating petri dishes and then placing small gallium pellets into the petri dishes to see if bacteria would grow on the gallium or not at all on the gallium. What I found was that while the bacteria grew fine, even with the gallium, the bacteria would not grow on the gallium, which may prove to show that the oligodynamic effect is in effect with gallium. The oligodynamic effect is where a metal will contain antimicrobial properties for a metal, such as aluminum, which is just above gallium on the periodic table, which prevents the growth of microbes such as fungus, bacteria, other different prokaryotes, etc. So I saw that gallium was absorbed into bacteria. However, it did not seem apparent that gallium was absorbed in the second stage of my experimentation. This can be further. This shows that it may not have been the most effective test I could have run. Further experiments would include mixing gallium nanopowder with the growth medium for these bacteria, so that perhaps maybe when the gallium is taken up as smaller minute particles into these bacteria's food, then maybe we will see some more drastic effects. So additionally, this can be used to further state that gallium has the oligodynamic effect in effect, and it can still be used as copper-gallium pipeline. However, tests still remain inconclusive for that matter. However, one fact we are sure about of gallium is that it discolors bacteria, making it darker. Additionally, observations from gallium when handled with gloves was that gallium would discolor the glove, and it could also be concluded that it would also affect eukaryotes in a similar manner of discoloration. If this has any more effects remains to be seen with further test studies. So the further research would be with gallium nanopowder mixed with growth medium to see how this affects further bacteria. Thank you.